So with that, we'll go into the review of our chemical foundations of biochemistry. So much of the basis of life is chemical reactions that take place in the cell. That's where the chemistry and biochemistry comes from. Chemistry is going to allow for that high degree of complexity and organization. As I just showed in the picture, the way that we can get this big organization and complexity is by small molecule building into larger and larger blocks. Chemistry also allows for things like extraction, transformation, and systemic use of energy to create structures and maintain what they do. So the actual chemistry, the reactions that are occurring are going to help us make energy to sustain life. Chemistry helps with things also like the interactions of components, the ability to sense and respond to changes, and a capacity for self-replication. In the review here, we're going to look at the chemistry behind the initiation and acceleration of reactions, organization and specificity of metabolism and signaling, and storage and transfer of information. Remember that organisms in general, we've classified them by their domains and their kingdoms, but they can also be classified more by the type of energy they use. You might have become familiar with some of these names like chemotrophs and phototrophs. These are names for types of organisms. They could be eukaryotic, they could be prokaryotic, they could range within the domains and kingdoms we talked about. But here we're naming them just based on what type of energy they use. So if they utilize chemicals to make energy and survive, we call them chemotrophs. And within that branch, there's actually even more specific types of chemotrophs depending on what type of chemical they use. And if they utilize light, we will call them phototrophs. And again, depending on how they utilize light, we can classify those even more into different types of phototrophs. Living systems in general extract energy, and energy is needed to maintain life. This is one of the main parts of living organisms and life that utilizes chemistry, is how we extract energy, right? How we extract energy from either sunlight, if you're plants, bacteria, or cyanobacteria, or from chemicals and fuels like we as animals or bacteria do, is one of the most important pieces of chemistry. We also utilize many different elements that are essential for life. There are, in fact, 30 elements in the periodic table that are essential for life. The main one being carbon. You guys have talked a lot about carbon in organic chemistry, and we're going to talk about it even more. It's one of the main building blocks of living organisms. Other elements that are very important are hydrogen, oxygen, nitrogen, phosphorus, and sulfur. But metal ions are also very important, and you're going to see that they play important roles in our metabolism and how we enzymatically complete different reactions. So metals are utilized a lot within us and us as animals and across living organisms. As I pointed out, carbon is probably the most important building block that we'll be talking about, and it's because it has a unique role within biochemistry. Carbon can form multiple types of bonds, and it can form bonds with other elements. And this particular component of carbon will be very important. This is a refresher to remind you that number one, carbon always makes 4 bonds. I drill this into my organic chem students. Always remember carbon's making 4 bonds to be happy. So we should never have carbon that has 6 bonds or has only 2 bonds. Something like a double bond counts as 2 bonds, and something like a triple bond counts as 3 bonds. So here you can see that's because carbon has 4 valence electrons, so it's going to form 4 bonds total. Here, if it forms bonds with hydrogen, this would be 4 single bonds with hydrogen. Or it can do something like this, where it forms a double bond that counts as 2, and it has room for 2 more bonds, still a total of 4. Carbon is also unique in its ability to form these nice triple bonds, which are utilized in living organisms as well. Now, when we're talking about carbon's special bond formation with other elements, we have to bring up functional groups. So you should have already seen functional groups in organic chemistry, and they will be brought up again here. As a reminder, functional groups are areas, they're types of specific groups made up of carbon and other elements, or sometimes they are just things like sulfur or phosphorus. But these special groups that we have designated that are very chemically reactive. That's why we focus on them, because they are usually the sites of chemistry that happen. They're usually the areas where we're seeing reactions take place. So here's kind of a list of general functional groups that are common in biological molecules. So a lot of these you should have already seen before, and if you've taken the pre-test, you would already been tested on your ability to kind of be able to distinguish some of these functional groups. Some of these might be a bit new to you, and they might be introduced through the semester as they are more specific to biological molecules and less seen in just general organic chemistry. But a lot of them you should know, like a general methyl group, an ethyl group, a phenyl group. This is that alternating 6-membered ring with double and single bonds. The types of carbonyls. This one is very important, and you should be able to distinguish, number one, what a carbonyl is. Remember, a carbonyl is all of the groups that have this carbon double bond O like this, and something here and something here. Depending on what these groups are on the side, it will be a different type of carbonyl. So for example, in our first one, an aldehyde is when you have a carbonyl that has a carbon on the left and a hydrogen. That doesn't have to be on the right, but it's bound to another carbon and a hydrogen. A ketone is when there is a carbon group here and here on each side. Carboxyl is where we have a carbon group here, and this is an O-. This can also be listed as a carboxylic acid. That's probably what you're most familiar with, and this is when this is an OH group, when that hydrogen is still attached. A hydroxyl or an alcohol group is just an OH group. This enol, now this might be a stretch for you to remember, but we did talk about ketoenolization. This is a tautomer. This is where you can go from a ketone to an enol form through resonance. Something you might want to review is how molecules can switch between ketone to enol. This is very important in biochemistry. You see this tautomerization happening a lot within molecules. Ethers, this is a carbon-oxygen-carbon bond, an ester, carbon, and then there's an oxygen here that is bound specifically to another carbon. Acetyls might be something that is new. This is, you can see this specific type of carbonyl group. And hydrides, where you have carbonyls that sandwich an oxygen. Amino groups are very important. We'll be seeing them a lot in our amino acids. And here are some on the right-hand side, functional groups that might be more specific that you might not have seen in your general organic chemistry, but that will be brought up within biochemistry, like guanidine groups, imidazole groups, disulfides and thiol esters are things that you'll be seeing a lot of as well. These phosphoryl groups and phosphoryl anhydrides are also very important. Again, we'll be seeing those in things like ATP and ADP. So review these functional groups. Your basic functional groups, again, you should be familiar with, but if I say something like disulfide bond, I want you to be able to kind of know what I'm talking about. So this is a very important chart that you should start reviewing. Within biological molecules, we actually have usually several functional groups. So I noted that these functional groups are sites of chemistry, and that's why they're important. But many complex molecules have multiple functional groups on them, multiple areas for chemistry, and sometimes there's one particular functional group that is more chemically active than others. But usually these functional groups are kind of building areas to elongate that molecule and make the final molecule. Now, I've talked about these small molecule building blocks, how they're made of functional groups, and how they make these more macromolecules. This is really the basis of biochemistry, and one of the areas we'll be focusing on a lot in this class, which is those simple ABCs, the building blocks of biological molecules and bigger structures, what they are and how they interact with each other. So the ABCs, the main building blocks we'll be talking about through the semester, are amino acids, which make up proteins. And I actually suggest that you start to memorize your amino acids now. We're going to get into that chapter and we're going to start reviewing them. But I've actually linked you a Quizlet on the left to start you now in your memorization process. Unfortunately, this is just something that you're going to have to memorize. Generally, what their structure is and their name. And it is important because we will be talking about their function, and you have to have some relative understanding of their structure to understand their function. We'll also be talking a lot about nucleic acids. This is one of the other main building blocks. Nucleic acids, of course, there are the main 5 nucleic acids, and they're sugars that they're attached to as well. So remember, nucleic acids are kind of built of these nitrogenous bases, uracil, thymine, cytosine, adenine, guanine, and the representative sugar that they are attached to, either ribose or deoxyribose, which again make up RNA or DNA respectively. And then the components of lipids. So lipids is the last of the ABCs that we'll be talking about. You can see that structurally they're very different. They're these long hydrocarbon chains with polar ends. There's also other portions of lipids we'll be going into, glycerols and choline, which are groups that attach to these lipids. One of the additional points of your ABCs to remember is we will be talking about carbohydrates and sugars. Sugars are still utilized very much in organic structures and biological structures. usually they are in tandem with one of these other main building blocks. They're either in tandem with an amino acid, in tandem with a nucleic acid, or in tandem with a lipid. They're still very important, and we still are going to be talking about their structure and relation. And then another guy that's very important and was brought up in our functional groups is the phosphate group. He's going to pop up a lot. Phosphate is used usually as a linker and for energy production. So just a quick question, kind of keep you on your toes. You just saw those main ABC molecules, the main building blocks we'll be talking about. So of the groups we've mentioned, which group of biologically important molecules did you note has physical properties that are very different from other groups, and thus its members also function very differently? So we're looking at either nucleic acids, carbohydrates, proteins, and lipids. Now, you might not know this exactly right now. We're definitely going to be diving into all of these molecules much more throughout the semester. But again, if you look back here, just kind of generally look at the structures that we see, the answer here is lipids. Lipids and fats are structurally very different than how nucleic acids, carbohydrates, and proteins function and even just look. Note that I said these are long hydrocarbon chains versus these usually rings of carbon, hydrogen, nitrogen, and oxygen that you see. All of our other components that we talked about, sugars, nucleic acids, and proteins, they usually work by linking together in polymers. Lipids don't do that. They're just already a long chain. Another key chemical point we'll be focusing on is the function of molecules, which is dependent on its three-dimensional structure. And when we talk about function and three-dimensional structure, we have to bring up isomers. Isomers are a key component of biological life. And to remind you, the different types of isomers are stereoisomers. Stereoisomers generally have different physical properties. Geometric isomers, so these are isomers that are things like cis versus trans. They have the same actual structure, chemical structure, but they are differently positioned, geometrically different. Enantiomers, remember, have identical physical properties except for the polarization of light, and they react identically with achiral reagents. These are any type of molecules that are mirror images of each other, remember, like our hands. And then diastereomers have different physical and chemical properties. Diastereomers are going to be similar to enantiomers, but they will not be mirror images. You will have some other type of rotation or movement when you compare the two molecules that make them non-mirror images. Here's a reminder on some of those types of some of those type of isomers. So here's something like malic acid and fumaric acid. These are two biological molecules that we utilize regularly in our mitochondria to produce energy. They are cis and trans molecules. Notice that they structurally are the same. This is a COOH group followed by carbon-carbon double bond and another COOH group. The only difference is whether the two bulky COOH groups, these carboxylic acid groups, are on the same side of the molecule or on opposite sides of the molecule. Remember, this is a cis bond versus a trans bond. Here's another example, and you can see the importance of it. This is retinol. This is a molecule that is utilized in the eye and also in other areas of the body. Retinol changes its function, and it actually changes its type of isomer between cis and trans when it encounters light. So you can see there is the cis version of retinol, 11 cis retinol, and when it encounters light, that bond between the 11 and 12 that is highlighted will then flip and form trans retinol. To refresh you on an enantiomer versus a diastereomer, remember in both of these cases, these are molecules that have the same structural layout. So as far as what is next to what, carbon next to another carbon next to an oxygen, is the same within these molecules. So they are not structurally different. They instead have changes based on the positioning around a component within the molecule. So you can see this is all different four-membered chains. So some type of butane that has some X and Y addition on it. You can see if the X and Y are either on the same side here or on the opposite side here. These are different isomers that are mirror images of each other. So this molecule and this molecule are enantiomers of each other. Similarly here, this molecule and this molecule are enantiomers of each other. Now, these are all isomers of the same molecule. And if we were to compare this molecule to this molecule, they are not mirror images, but they are isomers of each other. So these are diastereomers of each other. Similarly, this molecule and this molecule are diastereomers of each other. Again, this is a review of a concept you learned in organic chemistry, and it is an important concept to remember in biochemistry. So if you're still unfamiliar with difference between diastereomers and enantiomers, or what a geometric isomer is versus a structural isomer, these are all concepts to review now. Why do enantiomers versus diastereomers matter? So here is aspartame. This is L-aspartyl phenylalanine methyl ester versus the L-aspartyl D-phenylalanine methyl ester. So these are changed between each other. These are enantiomers of each other, mirror images, and you can see actually if I draw a line here that this molecule and this molecule are mirror images of each other. The positioning around that central carbon does make a difference. And you can see one is classified as D and one is classified as L around that carbon. And that small change in rotation makes one molecule sweet, which is the molecule that we call aspartame, and its enantiomer version bitter. Now, again, if you are feeling a little hazy on your stereochemistry, I've linked you some practice There is a video through Khan Academy to remind you of differences in stereochemistry, and there is also some practice questions if you're feeling like you just want to try to remember R and S and an enantiomer diastereomer. This interaction and stereochemistry is also important when we start talking about macro and molecules in 3 dimension. And one of the main reasons is when we're talking about binding sites within enzymes. We'll get into large proteins and their 3D structures. And most of these proteins have these binding pockets where they can bind small molecules. That's shown in the picture here. This is a large protein with a binding pocket, and a small molecule chemical has bound into it. And only certain molecules will be able to fit that binding pocket. Sometimes, and pretty regularly within organisms, including animals and ourselves, that binding pocket is actually specific to the chirality of the molecule. So even a small difference like we saw here, where it's mirror images of each other, the L versus the D and R versus S chiral center, it won't bond into that site. And this becomes very important, for example, when we talk about drug design. Pharmaceuticals and pharmacists are very, very concerned about chirality because of these specific, we call them stereo-specific binding pockets.